I'm Nidhi Kumar and you're watching Science Time, a show that brings to you the best that science offers, from exciting developments in science and technology to futuristic solutions. Pharmacogenomic landscape of Indian population identified. A computer made from DNA-coated beads could detect viruses in saliva. Forests help reduce global warming in more ways than one. Let's take a look at story number 1. Ethnic differences in pharmacogenomic variants have been well documented in literature and could significantly impact variability in response and adverse events to therapeutics. India is a large country with diverse ethnic populations of distinct genetic architecture. India's National Genome Sequencing Initiative provides a unique opportunity to explore the landscape of pharmacogenomic variants using population-scale whole genome sequences. The IndiGen team at CSIR IGIB, led by Sridhar Sivasubbu and Vinod Skeria, have analyzed the IndiGen variation dataset along with global population scale databases to map the most prevalent, clinically actionable and potentially deleterious pharmacogenomic variants among Indians. Differential frequencies for the known and novel variants were studied at the interaction of the disrupted pharmacogenomic genes affecting drug responses was analyzed by performing a pathway analysis. They identified 134 most common potentially harmful pharmacogenomic variants that could alter or inhibit the function of 102 pharmacogenes in Indians. They also estimate that on average each Indian individual carried 8 pharmacogenomic variants that directly impact the choice of treatment or drug dosing. The researchers have also highlighted clinically actionable pharmacogenomic variants and genes for which preemptive genotyping is most recommended for the Indian population. The study has put forward the most comprehensive pharmacogenomic landscape of the Indian population, from whole genomes that could enable optimized drug selection and genotype-guided prescriptions for improved therapeutic outcomes and minimizing adverse events. The work is published in the journal Clinical and Translational Science. And let's take a look at story number 2. Well, a type of DNA computer that shows results through the motion of tiny beads could massively increase the parallel processing power of such machines. DNA computers take up less space than silicon-based ones and can work in wet environments. They could be used for applications such as detecting contamination in drinking water or monitoring sugar levels in the body. Khaled Salatia and his colleagues from Emory University in Atlanta, Georgia, made the computers out of DNA-coated glass microbeads that either roll or stall on the surface of a gold chip depending on how the DNA strands interact with molecules attached to the chip surface. The results of this computation can be detected by tracking the motion of the beads using a smartphone camera with a simple magnifying glass attached to it. The team engineered guide DNA molecules onto the beads, which can bind to matching RNA molecules that are attached to the chip surface. The researchers then added a DNA lock complex into the computer, which enabled the presence or absence of a specific DNA molecule to control the movement of the bead. They confirmed that the presence of the DNA molecule stalled the bead, while its absence allowed the bead to roll. The system can easily be adapted to detect any DNA of interest in the environment. As there are thousands of possible microbeads with different shapes and sizes, the researchers say that the computers could output thousands of readouts in parallel. They hope the device could provide a quick way to detect virus levels in saliva. However, further work is needed to find out ways to restock the RNA molecules on the chip surface, which can degrade after 24 hours and limit the lifespan of the computers. The work is published in the journal Nature Nanotechnology. And with that, let's take a look at story number 3. When it comes to cooling the planet, forests have more than one trick up their trees. Tropical forests can help cool the average global temperature by more than 1 degree Celsius, a new study finds. The effect stems largely from forest capacity to capture and store atmospheric carbon, but around 1/3 of that tropical cooling effect comes from several other processes such as the release of water vapor and aerosols, researchers report in the March 24th issue of Frontiers in Forests and Global Change. Researchers already knew that forests influence their local climates through various physical and chemical processes. Trees release water vapor through their pores in their leaves, a process called transpiration, like human sweating. This cools the trees and their surroundings. Also, uneven forest canopies can have a cooling effect as they provide an undulating surface that can bump hot overpassing fronts of air upward and away. And what's more, trees generate aerosols that can lower temperatures by reflecting sunlight and seeding clouds. But on a global scale, it wasn't clear how these other cooling benefits compared with the cooling provided by forests capturing of carbon dioxide. The researchers analyzed how the complete deforestation of different regions could impact global temperatures using data gathered from other studies. For instance, the researchers used forest biomass data to determine how much of the release of carbon stored by those forests could perhaps warm the global temperature. Then they compared those results with other studies' estimates of how much the loss of other aspects of forests, such as transpiration, uneven canopies, and aerosol production, affected regional and global temperatures. And with this, we come to an end of this edition of Science Time. We'll be back with more exciting stories from the world of science next week. Until then, stay tuned to India Science. Namaskar and thank you for watching.